Okay, so where do cells come from? Pre-existing cells. Later we'll have to talk more about Francisco Redi and Louis Pasteur, and we'll go back through that. We'll start reviewing about scientific method. What are the three statements of the cell theory? All cells come from pre-existing cells. All cells are the basic unit of life. There is nothing smaller that is alive than a cell. All living things are made of a cell, because bacteria is a one cell or cells. Okay, so we're going to label this diagram, and I've already started doing part of it, so check yours. This part here is called mitosis. Remember this is all review for you guys. We say that the entire cell cycle takes somewhere between 23 and call it 25 hours. So the cells you have today, at this time tomorrow, you won't have. They will die, you will slough them off like your skin cells, and they'll be replaced by new cells. 90% of the time that a cell is in the cell cycle, it's in interphase. So 90% is in interphase. What's my saying when I say something about, if I'm writing it up here?
No, I was in the middle of number 5.
Pardon me? Can't say. No, that's not. Oh, that's number 5. Okay. It says, so we can label these. What's the first part of mitosis? Pimatzi. What's the first part? Prophase. Excellent. So we got prophase, metaphase,
Interphase.
okay, metaphase. We're going to go over these if you have any trouble spelling them. Anaphase, telophase. Okay, and what's the last one? So that's the part of mitosis. But we also say it's Pimatzi or PMAP plus C. What's C?
Cytokinesis.
Cytokinesis. C-Y-T-O. Cyto means cell. Cyto means cell. Kinesis means to move. So this is when the cells move apart. And it's the last time that that nuclear membrane is touching. And they're moving apart. That final division is happening. All right, G1, the cell grows. Nuclear membrane dissolves. Yes? Yeah, if you want to take this arrow here, what goes in this arrow?
Cell division.
It goes interphase.
No, but
Like which part? You can put anything you want in here as long as it's
no, let's see where the arrow's going and it leads
like this?
No, like inside the little triangle, like how it keeps the shape all at the end.
Yeah. That means this is just showing, Ryan, that it is a cycle. Oh, keep going there. Yeah, because that's what it shows in the book and on the book. So they actually break this out. So this says cell, as Ryan said, division. And we're going to get to why it says cell division right there. So G1, S phase, DNA is replicated. Replicated means an exact copy. So you replicate in a copier. If you go to a copier and you put your paper in there and you want 100 copies, every one of those copies is exactly the same. Okay? And that's really an important concept, like that these are exact copies. All right? So G2 cell checks to see if it's ready to divide, and organelles are finished. All this time the cell is growing. Once there, we go back into mitosis and we divide. Okay? Alright. I asked you to skip 4 and 5. Here are the steps in case you want them in Pimatzi. Prophase, metaphase, anaphase, telophase, and cytokinesis. What happens during cytokinesis? The last part before the two daughter cells are formed. Because remember, who can tell me what this is all about? We're going to get to it in our notes, but what is this all about? Mitosis. So we need cell division, but what are we trying to do?
More cells?
We're trying to make more cells. Excellent, Brian. But we're trying to make what kind of cells? Exact duplicates. You didn't even need the visual. We're trying to make exact duplicates.
Wait, isn't it during the side?
Cytokinesis?
Yeah, isn't it when two cells get one?
No, that's telophase. Telophase, it will split with the telophase, and we'll talk about that in a second, okay? Hang in there. I think all of this is going to make, and we're going to cover this both here and in our notes. Okay, so prophase. Some of you did really nice drawings and didn't write anything. Some of you wrote lovely things and didn't do any drawings. So let's see if we can get all of them. So I'm going to draw one time an animal cell. I'm going to draw a plant cell. What's the shape of a plant cell?
Rectangle.
Square, rectangle, good. Okay. Now, I'm going to draw a little rectangle inside of it. What's that? Cell wall. Okay, remember that this has a cell wall. We're going to come back to it and talk about that in a second. But up until I tell you, plant and animal cells divide exactly the same. I'll tell you when it differentiates. It's not till the end. So we're in prophase. Prophase. The nuclear membrane is starting to dissolve. Chromatin, which is the DNA inside the nucleus before prophase, kind of looks like you threw a bunch of spaghetti on a plate, okay? I kind of think it looks like steel wool. Well, you can't copy this, can you? I mean, you couldn't find two pairs in there to copy. So what it does is we start making the shapes that we're used to seeing, which are, so the chromosomes are going to become visible. The other thing that's really important, we're not going to need them much now, are these little guys down here, which are called the centrioles. So you could write in here the cell membrane dissolves or goes away. Chromosomes become visible. And I'm just going to write same here, because same thing's happening in the plant cell. Metaphase. What's happening in metaphase?
Chromosomes line up together.
Chromosomes line up together. M stands for middle. There's a couple of words I want you to know here. So I'm going to elongate, which means make just more of an egg shape, not much yet. Okay. Our centrioles, right here, have moved to, here's a word you have to know, poles. Here's a pole, here's a pole, just like the North Pole and the South Pole.
And we call the middle the equator.
And we call the middle the equator. Okay, so this is the equator. And then our chromosomes are going to line up here in the middle. They're going to meet at the middle. And our centromeres here are going to start shooting spindle fibers out. Okay? The central is going to send out spindle fibers. So chromosomes line up at the equator. Now, a question. So we've gone from 46 to 92. We're not going to have 92 long, because we're going to get on down the road here, and we're going to get the cell divided. So our cell's getting bigger, it's getting more oblong shape. Our centrioles higher. Our chromosomes are now getting pulled back. and this is anaphase. The chromosomes have been divided. So, let's see. We're going to ask Kevin. We started with 96 or 92 here. 92. How many are here now? Excellent. 46 are here. How many's over here now? 46. We're getting closer to having 2 daughter cells. All right. Telophase. This is the way I remember telophase. Telophase, you get cleavage. Now. So now our nuclear membrane is starting to come back. We're starting, the chromosomes are starting to turn back into chromatin. Our organelles are all in here. And this is called cleavage, just like in a human. Or just like my meat cleaver, which means to cut, cleave, to cut. Cleavage. So we have it in our chest, we have cleavage. We also have it in our bones, we have cleavage. But it means to divide, something that is divided. This is the way you can tell that this is telophase. Look for the cleavage. Okay? Now, I told you there was a difference with plant cells. Plant cells can't cleave. They can't do this in telophase. So what a plant cell does, and you must know this. You must know this. Put a big star here. You must know this. Animal cells are all soft and squishy on the outside. Our cell membrane outside of an animal is really soft, right? Our cell wall? Is that soft? No, we build our houses out of this. The outside cell wall is made of cellulose. That makes 2x4s and we build houses. So a plant can't do telophase. So what they do is they start building, you have to know the term of this, and they start in the middle and build out. They're building what is called a cell plate. And then when it connects, you're going to have 2 cells. Nucleus on both sides, same amount of chromosomes. Everything up to here, virtually the same. But it can't do this, so it has to do this. All right? We good? All right. We are done with this part. Give me a second here. Pardon me? Oh, we're going to watch it later?